beneficial interactions, but also of negative interactions such as parasitism and pathogenesis. Lactobacillus is an example of a benefic symbiont, and Criteria is an example of gut pathogen in bees. All the microbiota components are very diverse because they all perform different types of interactions. So how do bees acquire their gut microbiota? As previously mentioned, only a few bee species are completely social. Social bees have practices like trophallaxis, which is the transfer of food or other fluids among the members of the hive throughout mouth to mouth. Social bees have the transfer of microbes because of such practices. Therefore, the social bee microbiota is very constant. On the other side, solitary bee species acquire their gut microbiota through the environment by interacting with flowers. Thus, solitary bee microbiota is more variable as it is more regulated by the environment. Given that bees are extremely important for the pollination of our natural flower communities, and so they are for agricultural purposes, we're interested in understanding how flowers, bees, and their microbes and their symbiotic microbes change among different sites. There are two specific objectives for this project. The first objective is to assess community composition of the flowers, their bee partners, and their associated bacteria in 2 regions of Southern California. And the second objective is to compare the community composition and richness of species among the different studied sites. Community composition being a list of the species present in a certain time and space, and richness being the number of species. During spring and summer of 2021, my graduate student mentor collected bees in 2 sites that belong to the Santa Monica Mountains, Zuma Canyon, and Stunt Ranch. I participated in the data collection in Santa Rosa Island, but my grad student mentor collected also from Anacapa and Santa Cruz Islands. The islands have vegetation types of coastal sage scrub and chaparral. Both coastal sage scrub and chaparral are dominated by shrubs rather than full-size trees and are common in Southern California. So I was very fortunate to have the opportunity to experience doing fieldwork. In July, I traveled to Santa Rosa Island with my mentor to collect bee samples. While on the island, I observed several vegetation and used entomological nets to capture the bees. I learned to sweep the net and then flip it over the room to prevent the bees from escaping. The bees were then put into clean vials and were given a code for later identification. We searched for wildflowers and collected the bees interacting with them in plots of 2 square meters. We observed bees in each plot a total of 10 minutes, pausing the chronometer every time we collected a bee and processed it. We sampled the bees and the flowers in a total of 60 plots per studied sites. The collected bee samples were then stored in liquid nitrogen in a dry shipper on the island until we were able to store them in the -80 Celsius freezer on campus. As I mentioned, my grad mentor conducted 2 sampling rounds in April and July. However, I only participated in the fieldwork sampling in July, but I worked on analyzing with molecular techniques of all the collector samples from both rounds. After collecting our samples, we started working on bee gut dissections in order to examine for the microbes living in the bee's gut. Because we are interested in their microbes, we washed the bees in 1 bleach bath, 3 water baths, and they were dissected in a sterile environment to avoid contamination. After finishing the gut dissections of about 400 bees, we moved on to perform DNA extractions. DNA extractions were performed to isolate DNA from the nucleus of the cells. The DNA we obtained from the gut dissections was extracted by using a Quiagen DNeasy blood and tissue kit. We then began working on PCR with Illumina, and this would help us to perform deep sequencing in the 16S rRNA gene, which will give us all the bacterial symbionts present in the bee's guts. The PCR with Illumina protocol we are using consists of different steps, which are PCR1 to barcode the samples, a cleanup step, PCR2 to amplify the barcoded samples, and then another cleanup and normalization step. So we are still working on PCR, but once we have run PCR of all 5 plates of our samples, they will be sent to the genomics core for sequencing. We have consulted taxonomic keys along with internet resources such as iNaturalist, Discover Life, and Wildflower Search for plant identification. For bee taxonomic identification, we have observed bees under the stereoscope, and we have also used taxonomic keys. Here are our preliminary results. This graph represents the flower samplings that were collected in spring. In the x-axis, we have the study sites, Anacapa, Santa Cruz, and Santa Rosa, which are the islands, and Zuma Canyon and Stunt Ranch, which are the mainland. And in the y-axis, we have the blooming plant richness. Anacapa has 7, Santa Cruz has 16, Santa Rosa has 18, Zuma Canyon has 12, and Stunt Ranch has 8. 46 plant species have been identified so far. So that is more than half of the plants, but we still have others to identify, especially the ones from the July sampling. Even though all of the plants belong to coastal sage scrub and chaparral, so far we have seen that the studied sites shared few blooming species. For example, Santa Rosa Island and Santa Cruz Island only share 5 species in bloom for the April data, as you can see in this Venn diagram. These are the wildflowers shared among Santa Cruz Island and Santa Rosa Island from the samples collected in April. I still need to identify the ones from July, which will probably result in more shared species. Here we have Coreopsis gigantea, which is commonly known as giant Coreopsis, Calistegia macrostegia, which is commonly known as morning glory, Savia mellifera, which is commonly known as black sage, Lupinus albiforans, commonly known as silver lupine, and Escosia californica, commonly known as California poppy. And these are a few of the bees that I have been working with. Anthida melustra and Anthida manicatum are from the Santa Monica Mountains. They are from the same genus, but are different species. All Anthidium species are solitary, and they do not share the responsibilities of building a nest or providing for offspring. Then we have Serratina acantha. This is a small carpenter bee found in both Santa Monica Mountains and Santa Cruz Island. Serratina exhibit signs of sociality. While each female has her own nest, she keeps eye over the eggs she has laid until they develop into adults. Lastly, we have Bombus bonsanensky, which was also found in both Santa Monica Mountains and Santa Cruz Islands. They are among the few bees native to North America that are truly social, meaning that they live in a hive with a single reproductive queen and multiple sterile workers. As mentioned earlier, I still need to identify the rest of our plant and bee species, and also need to finish running PCR with the guidance of my mentor. Being in the RICE program and working in the McFrederick lab has allowed me to gain many transferable skills. I have learned how to be more organized and divide my time between lab work and other responsibilities. I have had more contact with scientific literature, and I was able to work with and learn about several protocols such as DNA extractions, and I was introduced to many machines and taught how to use them, such as the nanodrop to quantify DNA. I would like to thank all the amazing people that made this possible, especially the RICE coordinators, the 6-6 program, especially Dr. Jessica Purcell, and our research funding agencies. And last but not least, the McFrederick Lab, especially my PI, Dr. Quinn McFrederick, and my grad student mentor, Magda. Thank you for your attention. With this, I am happy to take any questions or comments.